Hey folks, here's video number 2 of this unit. It's called cellular respiration. Take a moment to write down the learning targets, please. Okay, cellular respiration is the catabolism of glucose into ATP. Catabolism is simply any chemical reaction that breaks down things. This happens in either with oxygen or without oxygen, depending on which kind of organism you're talking about. And some organisms like us, we do both. We do both with oxygen, without. Cellular respiration uses oxygen, so therefore it is aerobic respiration, and fermentation does not use oxygen, and therefore it's anaerobic respiration. The overall chemical equation shows that you take glucose and oxygen, you got to use the oxygen to catabolize the glucose, and that breaks down into 6 molecules of carbon dioxide, which are waste products, some water, and most importantly ATP. This is the entire reason that everybody does this. This equation should look familiar because it is the reverse. You take away ATP, it's the reverse of photosynthesis. So where this is a breakdown set of reactions, photosynthesis is a building up set of reactions. The parts of the mitochondria, which most of this occurs in, remember the mitochondrion is the double membrane organelle. It's got the outer membrane and it's got the inner membrane arranged in folds called cristae. So two of the three parts of respiration occurs inside the mitochondria. The 3 parts of cellular respiration are glycolysis, the Krebs cycle, and the electron transport chain. In this diagram, you can see that glycolysis occurs outside the mitochondria in the cytosol, whereas the other 2 processes occur at different locations inside the mitochondria. This is why the mitochondria gets the nickname the powerhouse of the cell, because it makes energy, like ATP. So step number 1, glycolysis. This literally means to split glucose. So glucose is going to be divided into 2 3-carbon molecules called pyruvate. So for every 1 glucose, you get 2 pyruvate. That is the end result of glycolysis. Along the way, though, you make 2 ATP. You actually have to spend 2 ATP to get this reaction started, but then you end up making 4 more ATP. 2 plus 2 here is 4. And so you have a net gain of 2 ATP. Not much energy, it's not enough for us to live on as human beings. This reaction also produces 2 NADH, which are energy carrying molecules that will be used later on in the 3rd step. So remember that the 2 pyruvate are the main products of glycolysis, and this is what moves on to the 2nd step, which is called the Krebs cycle. The Krebs cycle occurs in the matrix of the mitochondria. The matrix is that fluid-filled portion in between the folds of the inner membrane called the cristae. This is very complex, and this does leave out many steps. So for every 1 pyruvate that comes into the Krebs cycle, we produce 2 ATP, again, not much. Most importantly, 6 NADH and 2 FADH2. Now, FADH2, for this purposes of this class, let's treat that just like NADH. It's an energy carrier molecule. So for every 1 pyruvate, we get 1 ATP, 3 NADH, 1 FADH2, and 2 waste products called CO2. So this misspoke, I'll go for every 2 pyruvates, which comes from 1 glucose, we get a total of that. Let me go back to that. So for both pyruvates, we get 2 ATP, 6 NADH, 2 FADH2, and 4 CO2. So it's these 2 here in the middle that are the most important. They're the ones that's going to go on to the electron transport chain to be converted to even more ATP. So just remind you that NADH carries hydrogen ions and electrons similar to NADPH in photosynthesis. It kind of has the same role. So finally, the electron transport chain. This is the most complex, but I'm leaving out most of those complex steps. So it's going to take all those electron energy carriers like NADH and FADH2 produced in the previous 2 processes, and it's going to use oxygen to convert all that into lots and lots of ATP. So far, if you think about it, we made 2 ATP from glycolysis and 2 ATP from Krebs cycle. So we're going to end up making 32 ATP alone just in the electron transport chain. The movement of these hydrogen ions that NADH and FADH2 are carrying, they're going to be pumped across the membrane, and this is going to generate enough energy to turn ADP into ATP. As I said, 32 of them. So I don't really expect you to get much from this diagram because it is quite complex, but the gold path kind of shows you the flow of electrons and hydrogen ions. So hydrogen ions are dropped off of these two molecules. As you can see here, they're losing their H's. They move across the membrane, and they're going to diffuse back through this special enzyme called ATP synthase, and that's going to provide enough energy to convert ADP into ATP. And as I said earlier, it's going to actually provide, it's going to make 32 ATP. So here's a summary, here's a graphical summary of everything that's happened so far. Glycolysis occurs in the cytoplasm. It's going to produce 2 NADH, which goes on to the electron transport system. It's going to produce 2 ATP directly, and most important, it's going to produce 2 pyruvate. This is also known as pyruvic acid. These are going to go on to the mitochondria, where they're going to generate, in the Krebs cycle, a lot more energy carriers, 6 more NADH2s, sorry, 6 more NADH, 2 more FADH2s, actually 4 more if you count these two. It's also going to produce 2 ATP, so far we got a running total of 2 plus 2 ATP, and then this makes 34. I said 32 earlier, but remember you used up 2 over here to get this whole thing started. So if you take 38, and you take away the 2 that you used to get glycolysis started, that was the energy input, then you end up with a net of 36 ATP from 1 glucose molecule. All right, so what happens if there's no oxygen available? Or what happens if you're an organism that doesn't need a lot of ATP, like a yeast cell or a bacterial cell? Then you're gonna make all your ATP anaerobically because you don't need a lot of ATP. So here are the learning targets for fermentation or anaerobic respiration. Take a moment to write them down, please. So anaerobic respiration is simply the production of ATP without oxygen. This is the method used by plants, yeast, and bacteria. There's 2 types of fermentation. By the way, like I said, anaerobic respiration is also known as fermentation. Lactic acid fermentation, which is what animals do, and some bacteria does. And alcoholic fermentation, which is what yeast and some plants do. All right, so the diagram is this here, okay, where you're going to start off with glucose, which is what we start off using aerobically. It's going to break down into 2 NADH, 2 CO2, 2 ethyl alcohols, and 2 ATP. Now, of course, the ATP is what the yeast or the other organism is needing for their own body to keep it going, for their own cell. The ethyl alcohol is a byproduct, and the CO2 is a byproduct. So the 2 pyruvate that come out of glycolysis, this includes glycolysis, is going to actually be used to create the rest of this. So this organism does glycolysis just like animals like us do glycolysis. So what are some uses for alcoholic fermentation? Well, making dough that rises because of yeast being added to it is one idea. So when you mix in yeast with flour and water and sugar, the sugar provides enough food to feed the yeast. The yeast consumes the sugar, does alcoholic fermentation, and it gives off ethyl alcohol, which is that kind of a sweet smell when dough rises in your kitchen. And it also provides the CO2 bubbles, which lifts the dough up, and this makes the dough rise. Of course, when you bake the dough into bread, the alcohol is evaporated away, and the CO2 bubbles become spaces in your bread. When you slash your bread, those little tiny holes in your bread, that's because of the CO2 bubbles. The second type of fermentation is called lactic acid fermentation. Now, at some point during exercise, humans do this. But most importantly, some bacteria do this. Bacteria that's used to produce cheese and yogurt, bacteria that's used to produce kimchi, which is Korean pickled cabbage, sauerkraut, a German cabbage dish, sourdough bread, uses lactic acid. So this is very prevalent in the food industry. And it's very much like alcoholic fermentation. Let's go back to that. Except there's not going to be CO2 and there's not going to be ethyl alcohol. Instead, it's going to be lactic acid. So it's going to produce 2 NADH, 2 lactic acid, and 2 ATP. Now, that's going to give things like cheese and yogurt and sourdough bread their tangy taste, because lactic acid, of course, is an acid, and acids, one of the properties of acids is it has a sour taste to it. So you can see here at the bottom the actual equation for lactic acid fermentation. Now, we do this, humans do this, when we're doing extreme exercise, and we've already basically maxed out our aerobic respiration system, and then this system kicks in and tries to provide us a little bit more energy, only 2 ATP, of course, though. And when the lactic acid starts to build up in the muscle groups that you're working so hard, then that causes a burning sensation. Have you ever heard someone say "feel the burn," or ever heard of or you've ever done burnouts in the weight room? You basically have exhausted your muscle, exhausted all your ATP, and your muscle have lactic acid build up inside of it. So that's what causes the burn. So humans do lactic acid, bacteria do lactic acid fermentation. That's it. Thank you for watching. Please be sure you take good notes. Our notebook check will be on Monday the 15th. Thanks.